Okay, so welcome back to this next video in which we are discussing cholesterol metabolism. Okay, so we've just been discussing the exogenous pathway, which is this way of delivering cholesterol from the enterocytes to the hepatocytes. But we've seen that really it's an afterthought, the delivery of cholesterol to the hepatocytes. Really, the principal part of the exogenous pathway is the delivery of triacylglycerols from the enterocytes to the skeletal and cardiac muscle cells and the adipose tissue. Okay, and then the remnants of the chylomicrons afterwards can then be endocytosed by hepatocytes and thereby deliver dietary cholesterol to the hepatocytes. Okay, we're now going to turn our attention to the endogenous pathway, and the endogenous pathway is very similar. Okay, really the endogenous pathway is about the hepatocytes which have excess triacylglycerols, sending these triacylglycerols to the skeletal and cardiac muscle cells and also to the adipocytes. And then as an afterthought, the cholesterol is added into the LDL pool. Okay, so let's now study then the endogenous pathway. So the endogenous pathway, like the exogenous pathway, is really only going to be active in the fed state, okay, because it relies upon the hepatocytes having excess triacylglycerols. Okay, fundamentally this pathway is about the hepatocytes getting rid of triacylglycerols that they have synthesized. Okay, now why do hepatocytes produce triacylglycerols? Okay, well they produce triacylglycerols in the eventuality that they have far too much of certain nutrients. Okay, so when you have just eaten, you often eat more nutrients than actually is needed. Okay, so for instance, you will consume too much glucose, more glucose than is actually needed to fill up the glycogen reserves. Okay, so once the glycogen reserves in hepatocytes and also in skeletal muscle cells are full, the rest of the glucose is now excess. Okay. In addition, often you'll end up with excess amino acids, the constituents of proteins. Okay, so when these two nutrients are in excess after a meal, okay, what's going to happen is the hepatocytes are going to use these nutrients to manufacture triacylglycerols. Okay, so they manufacture fat molecules from these excess nutrients, basically. Okay, and this is occurring in the fed state. The hepatocytes use the excess nutrients to produce triacylglycerols. And now what do they do with the triacylglycerols? Well, they have to send them to tissues that can actually either use them or store them. So they're going to send them to skeletal and cardiac muscle tissue and also adipocytes for storage, basically. Okay, so once again, how do you carry triacylglycerols from one portion of the body to another portion of the body? Well, you require a lipoprotein to do that. Okay, and the lipoprotein that's going to be important in the endogenous pathway in carrying triacylglycerols from hepatocytes to these other tissues is going to be another new one, okay, which is very low dense lipoprotein. Okay, so VLDL. Okay, so let's start off with a discussion of VLDL, okay, because we haven't discussed this one in detail yet. Okay, so when we were having the general discussion then on lipoproteins, I told you that VLDL is the second biggest, okay, after chylomicrons. They can be between 30 and 80 nanometers in diameter, so they are large lipoproteins. Okay, nowhere near as large as 1,200 nanometers, which is what the chylomicrons could get up to, but they are still big lipoproteins, certainly compared to HDL particles. Okay, now, the other key pieces of information that I previously gave you about VLDL particles is the apolipoproteins present in these VLDL particles. So let's start off with the big E, okay? So the big apolipoprotein that they contain is the apolipoprotein B100. That one that we saw is the only apolipoprotein present in LDL, okay? and that's a massive grade apolipoprotein. And indeed, we will see that these VLDL particles are eventually going to become LDL particles, and this apolipoprotein B100 that you have here in the VLDL particle is going to be the apolipoprotein B100 that's going to be eventually in the LDL particle that will form from this VLDL particle. Okay, now, VLDLs, however, have other important apolipoproteins beyond apolipoprotein B100, and another example of an important apolipoprotein, which I'll put here, is the apolipoprotein C2, okay, which we've met before. Okay, remember that one is the one that activates lipoprotein lipase. So this is all starting to come together. Remember, what's the function of these things? It's going to be mainly to carry triacylglycerols to muscle tissue and also to adipocytes. Okay, so you might be starting to put this all together as to how the triacylglycerols are going to be unloaded at those sites. Okay, it's going to be in the same way as the triacylglycerols of chylomicrons was unloaded at those sites. Okay, right. So they are similar to chylomicrons, okay? They will have a phospholipid monolayer which will consist of phospholipids and a few odd cholesterol molecules dotted around the place, okay? And their core is going to have a lot of triacylglycerols in it, okay? So here are the triacylglycerols represented here. So once again, I'll colour in this picture. So here are the long-chain carboxylic acid molecules attached to the glycerol molecule here in green. Okay, and you will also have cholesterol esters in the core as well. So I'll put one of these in here. So here is the long-chain carboxylic acid in orange there, and then here is the cholesterol molecule in red. Okay, and that's a cholesterol ester there. Okay, right. Now, they're not quite as extreme as chylomicrons. The triacylglycerols only make up about 50% of the core. Okay, so it's not the 90% that was the case in the chylomicrons. Okay, right. So VLDL then, they're going to be secreted by hepatocytes when you're in the fed state, and they're being secreted because the liver has a lot of these triacylglycerols which it has been synthesizing from excess nutrients such as glucose and amino acids. Okay, they're also going to contain lots of cholesterol esters. Again, the liver's received lots of cholesterol from dietary sources. Okay, so some of that will end up being packaged into these VLDLs. But understand that the main function that these things are performing is to get rid of the triacylglycerols. Okay, the removal of the excess cholesterol in the hepatocytes is almost the afterthought. Okay, right, so now these things are going to circulate in the blood, they're going to go into capillaries in muscle tissue and also in adipose tissue, and there what's going to happen is exactly the same as what happens in the case of chylomicrons. So if I just get the picture back, I would be basically drawing the exact same picture again. Lipoprotein lipase is going to stick its head into the VLDL particles. Apolipoprotein C2 is going to bind to lipoprotein lipase and activate it, and then lipoprotein lipase is going to break down the triacylglycerol molecules into glycerol molecules and free fatty acids. These will diffuse out of the VLDL lipoprotein particle. They'll diffuse across the endothelium of the capillaries into the tissue, whether it's muscle tissue or adipose tissue. In muscle tissue, those molecules will then be used for metabolism, okay, by beta oxidation of those fatty acids, okay, which will produce acetyl coenzyme A, which can go through the Krebs cycle and then drive the electron transport chain. And in adipocytes, what will happen is the glycerol and the free fatty acids will be reassembled to produce triacylglycerol once again, and that can be stored by the adipocyte. Okay, right, so once again what's going to end up happening is these VLDLs are going to be gradually depleted of their triacylglycerol. Okay, so their core is going to deflate, basically. They're going to lose volume from the lipid core, okay? Now, that means they need to lose surface area, basically. So again, they will lose phospholipids to HDL particles and apolipoproteins to HDL particles. Okay, now as they gradually shrink, they go through two phases, okay? So as they shrink, they become what is known as an IDL particle, okay? And then after they shrink even more from being an IDL particle, they then become an LDL particle. Okay, right, so let me distinguish between IDL and LDL. Okay, so IDL is smaller than VLDL. Remember, IDL stands for intermediate density lipoprotein. Okay, and the diameter of IDL particles is between 25 and 35 nanometers, so smaller than the VLDL particles. So this diameter is now 25 to 35 nanometers. Okay, and the major thing that distinguishes IDL from LDL is that IDL will still have more apolipoproteins than just the apolipoprotein B100. Okay, so let me draw this here. So you will obviously still have the apolipoprotein B100, but you won't yet have lost all other apolipoproteins other than that one. Okay, to become an LDL, you have to have lost all other apolipoproteins other than the apolipoprotein B100. Importantly, IDL will still have the apolipoprotein C2, at least some copies of apolipoprotein C2. It might have lost a few, but it will at least still have some. Okay, it will have less, obviously, triacylglycerol in its core than the VLDL, but it will still have a bit there. Okay, so you'll have been depleted, but you're not yet out of triacylglycerol. So basically, these IDL particles can still shrink further, is the key thing here. So let me just colour in these components. So there are the long-chain carboxylic acids of the triacylglycerol. There's the glycerol molecule in green. And here is our cholesterol ester here, cholesterol in red, and the acyl group in orange. Okay, so IDL is the shrunken version of VLDL. Okay, but you still have some triacylglycerol molecules in that lipid core. Okay, and you still have more than just the apolipoprotein B100 apolipoprotein, basically. Okay, so you have other apolipoproteins still in the phospholipid monolayer of the IDL particles. Then finally, we know that this can still be depleted further, so more triacylglycerols can be taken out of this. You can lose more phospholipids, more apolipoproteins, and then you'll shrink down to this smaller particle, the LDL particle down here, okay, which has a diameter between 18 and 25 nanometers. Okay, so now we're getting to a smaller diameter here, 18 to 25 nanometers, and now the only apolipoprotein left is that apolipoprotein B100, which dominates the LDL particle. Okay, and really you've now lost all of your triacylglycerols from the core, so really what you have left in the core is a lot of cholesterol esters here. Okay, like so. So here's the cholesterol molecule, and here's the acyl group in orange. Okay, right, so that's how VLDL particles can gradually, through the IDL particles, become LDL particles. So it's a gradual transition from VLDL into IDL into LDL by the loss of triacylglycerol from the core, which then triggers loss of phospholipids from the phospholipid monolayer and the loss of apolipoproteins from the phospholipid monolayer. Okay, so if you like, the IDL and the LDL are really the remnants of VLDL molecules, okay? Right, okay, so that now is the endogenous pathway completed. Okay, this is how the liver produces these VLDLs, which supply triacylglycerols to the muscle tissues and to the adipocytes. Okay, and then the remnants of these VLDL particles, after they've lost the triacylglycerols, they first transition into this intermediate density lipoprotein, and then finally they end up becoming these LDL lipoproteins, okay? And now the cholesterol that hepatocyte originally put into these VLDL particles has now been put into the LDL pool within the blood. Okay, so that now is our cholesterol metabolism finished. Okay, what we now want to look at is some important corollaries of this pathway. Okay, so we want to now talk about hypercholesterolemia. Okay, specifically we'll look at familial hypercholesterolemia and then diet-induced hypercholesterolemia. And we want to look at the very, very severe consequences of hypercholesterolemia, which is cardiovascular disease, which results because of atherosclerosis, which is triggered or at least made worse or accelerated by hypercholesterolemia. Okay, right. So firstly then, let's discuss this big word, hypercholesterolemia. Okay, so it means too much cholesterol in the blood, basically. Hyper means too much. Okay, cholesterol obviously refers to cholesterol. Emia means level in the blood, so too much cholesterol in the blood is what is meant by hypercholesterolemia. Okay, and in hypercholesterolemia, quite simply, you have too many LDL particles in the blood. Remember, LDL is the principal store of cholesterol in the blood. Okay, so the main way that you can get too high cholesterol levels within the blood is by having too high LDL levels. Okay, so too many LDL particles within the blood is therefore going to cause too high cholesterol level within the blood. Okay, and that's what is usually meant by hypercholesterolemia, too high LDL level within the blood. Okay, right, so let's now talk about familial hypercholesterolemia, which is an inherited disorder which can lead to you having too high LDL levels. And then what we'll talk about is diet-induced hypercholesterolemia, which is where having a too high cholesterol diet is going to induce the hypercholesterolemia, the too high LDL levels within the blood. Okay, so let's start off with familial hypercholesterolemia, which for short is abbreviated to FH. So I suppose I probably should write it out in full. So the F is for familial, meaning that it runs in the family. Okay, and then the H is for hypercholesterolemia once again. Okay, so basically familial hypercholesterolemia results because of germline mutations in the LDL receptor. Okay, and these germline mutations in the LDL receptor are going to lead to the LDL receptor not functioning properly. Now, why is this going to lead to LDL getting too high? Well, remember, how do you remove LDL from the blood? It's endocytosed by LDL receptor mediated endocytosis. That's the principal way in which LDL is removed. If you have then mutations in the germline in your LDL receptors, then this is going to be a mutation that's in every single cell of your body, basically. Okay, and then cells are not going to be able to endocytose LDL as well. Of course, it will depend on whether you're a heterozygote or a homozygote, which we'll come on to as to how severe this becomes. But you can understand now why that's going to lead to LDL growing and growing and growing in the blood, basically. Okay, because you're still going to be putting LDL into the blood through, for instance, the endogenous pathway and reverse cholesterol transport. Okay, but you're not now going to be taking it out of the blood, so it's going to build up and up and up in the blood. Okay, so let me firstly then talk about mutations that can occur in the LDL receptor, because they're grouped into categories, basically. Okay, so in familial hypercholesterolemia then, you're going to have loss of function, LOF, mutations in the LDL receptor. Okay, and these loss of function mutations in the LDL receptor are classified into 4 classes, okay, and I want to discuss these 4 classes with you now. Okay, so let's start off by discussing class 1 mutations in the gene for the LDL receptor. Okay, so in class 1 mutations in the gene for the LDL receptor, you get some mutation, some permanent change in the sequence of organic bases in the gene for the LDL receptor, and this leads to absolutely no LDL receptor being synthesized. Okay, so these are usually nonsense mutations. They usually lead to a premature stop codon being created, and therefore the translation of the mRNAs for these LDL receptors end up being terminated far too early, okay, so end up producing no functional protein. Okay, so in class 1 mutations of the LDL receptor gene, then that gene cannot actually synthesize any functional LDL receptor whatsoever. Okay, now as I say, we'll discuss heterozygotes and homozygotes in a moment, because of course how severe this is going to be for you is going to depend on whether you're a heterozygote or a homozygote, but we'll come to that. Okay, at the moment we're just discussing mutations in a single gene. Okay, so a class 1 mutation occurring in the LDL receptor gene means that that gene is not capable of synthesizing any functional LDL receptor whatsoever. Okay, then there are class 2 mutations in the LDL receptor gene. Now in class 2 mutations, you do synthesize a protein that looks reasonably like the LDL receptor, but unfortunately what happens is it slightly misfolds. Okay, so the protein doesn't adopt its normal three-dimensional conformation. It misfolds basically, and then something about the misfolded protein is recognized in the ER. Okay, so let me just go over how the LDL receptor is actually moved to the plasma membrane. Okay, so if I just draw a little picture of a cell here, okay, we'll have the nucleus here, here's the inner nuclear membrane, here's the outer nuclear membrane continuous with the endoplasmic reticulum. Now basically the LDL receptor is going to be folding in the endoplasmic reticulum. All membrane-bound proteins fold in the endoplasmic reticulum. So here we'll have the LDL receptor folding in the endoplasmic reticulum. So here it is, like so. Here's its ligand binding domain here. Okay, then what will happen is, to get it to the plasma membrane, normally what has to happen is you send it firstly to the Golgi, so this is representing the Golgi here. Okay, so first it will be moved to the Golgi. Here it is now in the Golgi, and that will occur just by a vesicle moving from the endoplasmic reticulum to the Golgi. Okay, so this is the ER. And then what will happen is it will move through the Golgi, and then again a vesicle will transport it from the Golgi to the plasma membrane. Okay, so that's the normal way that it should be trafficked through the cell to the plasma membrane. Now, if you've got a class 2 mutation in the gene for the LDL receptor, what ends up happening is it never leaves the endoplasmic reticulum. Okay, the protein is recognizable, okay, so it looks reasonably normal, but something about the protein is slightly misfolded, and quality control mechanisms in the endoplasmic reticulum do not let it go to the plasma membrane. Instead, they degrade it. So it slightly misfolds, and then it gets degraded, basically. Okay, so you don't actually get any functional protein from that gene getting to the plasma membrane. Okay, so you might as well have not bothered synthesizing it in the first place, because it's not actually getting where it needs to actually function. So that's what occurs in a class 2 mutation in the LDL receptor. Then let's talk about class 3 mutations. In class 3 mutations, the LDL receptor does get to the plasma membrane, okay, so it doesn't misfold, it does get to the plasma membrane, okay, but when it gets to the plasma membrane, it doesn't bind LDL, okay, so the problem is with the binding domain, okay, problem is in binding LDL, it doesn't do it anymore. Okay, so it's got much less ability to actually bind LDL, so some mutations occurred in the ligand binding domain that means now that it's just not capable really of binding LDL at all well. Okay, and therefore effectively again it's a useless LDL receptor. We've got it there to the plasma membrane, we've synthesized it, we've trafficked it there, but it's not actually functional. Okay, finally, and I don't know why I've suddenly moved to using Roman numerals rather than the Arabic numerals. Okay, but never mind, that's class 3. Okay, next up, class 4. Okay, and I'll switch back to using more normal numbers. Okay, at class 4 then, finally, in class 4 mutations, the LDL receptor gets to the plasma membrane, it can bind LDL, but now there is some problem in that sorting motif, okay? And I mentioned this at the time, NPXY motif, that key motif for the LDL receptor to bind to the adaptor protein complex 2, okay? And that was absolutely key for the LDL receptor with the LDL bound to actually be endocytosed, because that's what fastened the LDL receptor in these regions of the plasma membrane which were being endocytosed. So I'll just try and find a picture. Okay, so here we go. So remember, in order to get your LDL receptor to localize in these portions of plasma membrane which are being endocytosed, you need this sorting motif here, because this sorting motif is what binds to the adaptor protein complex 2. Okay, so it's very essential that that sorting motif is intact for you to actually get a decent amount of LDL actually being endocytosed. Okay, and in class 4 mutations in the LDL receptor, you end up with problems in that sorting motif, which mean that it no longer works, it no longer binds to the adaptor protein complex 2, and that also severely disturbs the function of the LDL receptor. Okay, so now it can bind LDL, but it doesn't actually get endocytosed. Okay, so these are the 4 classes then of mutations in the LDL receptor gene, okay, which can then lead to familial hypercholesterolemia. Okay, now, this is all well and good. This is about a single gene, but of course, in humans, in each one of our cells, we are going to have 2 copies of the gene for the LDL receptor, because after all, it's on an autosome, it's not on one of the sex chromosomes. Okay, so that then leads to the possibility for you to either be a heterozygote for familial hypercholesterolemia, to have 1 normal copy of the LDL receptor gene and 1 mutant copy of the LDL receptor gene, or for you to be a homozygote. So let me just talk through this. Okay, so here are 2 chromosomes with LDL receptor genes on, and I don't actually know the specific number of the chromosome 1 to 22, which one the LDL receptor is actually on, but the important principle is that it's on an autosome rather than a sex chromosome. Okay, right, so let's say that this portion in blue here, this represents the LDL receptor genes. Okay, so here is our maternal chromosome with the LDL receptor gene here, and this is our paternal chromosome with the LDL receptor gene here. Okay, so either you can be a heterozygote for familial hypercholesterolemia, okay, which means that one of your LDL receptor genes is normal. So let's say the maternal one's normal and the paternal one is then faulty. It's got one of these mutations in, one of these classes of mutations that leads to loss of function of the LDL receptor. Okay, usually heterozygotes are affected, okay, they're not asymptomatic, okay, they end up usually with twice as large LDL levels within the blood as is normal, basically, okay, so they do end up hypercholesterolemic. Now, what's the effect of this? Well, generally people who are heterozygotes for familial hypercholesterolemia die of cardiovascular disease, so heart attacks and strokes, 10 years earlier than would be normal, basically. Okay, so they are generally people who die of cardiovascular disease prematurely, but it's only premature by 10 years, and that will seem okay compared to what the homozygotes are about to suffer. Okay, so they get cardiovascular disease 10 years early. Okay. Right, so, and we'll discuss exactly why that is. We'll discuss why hypercholesterolemia is going to cause cardiovascular disease, strokes and heart attacks in just a moment when we come on to atherosclerosis. Okay, right, so that's the prognosis for people who are heterozygotes for mutations in the LDL receptor. Now let's talk about the poor homozygotes. Okay, so if you're a homozygote, and I should stress that homozygotes for familial hypercholesterolemia are very rare. Heterozygotes are not as rare as you would think. Okay, homozygotes are really around 1 in a million. Heterozygotes are 1 in 500 generally. So heterozygotes are more common than you would like. Okay, so homozygotes then now. So these are very rare. Okay, so people who are homozygous for mutations in LDL receptors that cause loss of function, so both genes have now got mutations in, and that means that we have no functional copy of a gene for the LDL receptor anymore. People who are homozygotes for familial hypercholesterolemia, generally they have 5 times the level of LDL as is normal. Okay, and they generally don't ever get to their 20th birthday. Okay, they generally die of cardiovascular disease before the age of 20. Okay, so they'll have strokes or heart attacks before the age of 20. Okay, cardiovascular disease before 20 years old. Okay, so it's not a nice prognosis for familial hypercholesterolemia if you're a homozygote. Okay, right. So what I now want to discuss then is, firstly, I want to go on to diet-induced hypercholesterolemia, and then I want to discuss why does hypercholesterolemia actually lead to cardiovascular disease. Okay, so we'll discuss the pathogenesis then of atherosclerosis. Okay, so firstly let me just discuss diet-induced hypercholesterolemia. I think I'll get a new piece of... we can go on to the other side. Okay, so diet-induced hypercholesterolemia. Okay, so it's not just people with familial hypercholesterolemia that have far too high levels of LDL. If you eat a very cholesterol-high diet, then you can end up with far too high levels of LDL as well, and this can put you at risk of developing cardiovascular disease because of atherosclerosis, which we're about to discuss. Okay, so diet-induced hypercholesterolemia. Okay, so what sort of foods then are high in cholesterol? So it's things like butter, cheese, fatty meats. These are all things that are going to be very high in cholesterol. Okay, and if you eat too much of those, then you're going to be having a too large supply of cholesterol. Okay, and of course we know what happens to this cholesterol. It goes to the liver, and then the liver puts it into LDL through the endogenous pathway. Okay, one of the main things that happens. Okay, so you're going to be getting far too much LDL being produced, more LDL then than hepatocytes and peripheral tissues are going to be taking out through LDL receptor mediated endocytosis. Okay, and that means that your LDL level is going to start to rise basically within your blood. Okay, right, so that's how you can become hypercholesterolemic through bad diet, basically, too much high cholesterol foods. Okay, and a lot of people have now higher levels of LDL than is healthy because of bad diet. Okay, right, so we'll call it there for this video, and in the next video what we will move on to is discussing atherosclerosis pathogenesis, okay, which is the link between how hypercholesterolemia, either through bad diet or through familial hypercholesterolemia, can actually cause increased cardiovascular disease, heart attacks and strokes.